Good morning, sir.
Good morning. Good morning, students.
Good morning, sir.
Good
Good
morning, sir.
morning. Okay, students, that in the last period that we had finished that of yes, Tanushree Bhag, good morning. Yes,
Are not visible, sir.
yes,
Sir, your
not
camera is
audible.
no, sir. Your
Any
camera is
problem?
blurred, sir.
Yes,
Sir, camera is blurred. Sir.
hello, students.
Yes, sir. Sir, your camera is not visible at all.
Okay, that what problem with it?
Blur, sir.
Now visible?
No, sir. Whiteboard is blur.
Uh,
Sir, clean your camera.
light on. Now visible, it is possible or not?
Sir,
No,
please
sir.
clean your camera.
Yes,
Now
sir. Now,
students visible?
yes, sir. Yes, sir.
Yes,
Yes,
sir.
sir.
Okay, you try. That means only one period left. That means you'll have to go to that offline class. Okay. Okay, you see that. That means in the last period that we had discussion about that of light reaction and we had completed it, and in chemi osmotic process in hypothesis how you didn't know about that of ATP synthesis. Okay, ATP synthesis occurred, and you know that how that proton gradients, and that means maximum amount of protons in that of lumens, and it brought about that of and that lumens, and from that of that is proton ions that remove from that of lumens and come out to that of stroma, and by the process through that of ATP, that is synthesis complex, and in that ATP synthesis complex there are two parts. One is that of CF0 and another one is that is CF1. In that of CF1, ATP synthesis enzymes are present which have to synthesize that of ATP. And you have known that all these things about that of light reaction. And in the light reaction, we have known that there are three products, and one is that of assimilatory power. Two are called assimilatory power, that are ATP and NADPH. And another one is that of oxygen. Okay, that is oxygen, molecular oxygens, and that you use in form of that of in our respiration, or it exists to that of atmosphere. Okay, in such a way that we have know about that of light reaction. So that of light reaction, you know that it is that of you see that. Okay, you see that it is that of a mesophyll cell. In that of mesophyll cell, you can have to find out that of chloroplast, and in that chloroplast, that is that of that is thylakoids. Okay, that thylakoids. These are that of thylakoids, and these are that of and these are that called that is stroma. Okay, stroma, these are that of thylakoids. Okay, and in that thylakoids, that you know that is process, that is that is photosynthesis occur. Photosynthesis occur, and in that process, what you get, that means you see that suppose this is two part. This is that of stroma, and this is that of a thylakoid part. And here you will have to get that of NADPH and also ATP and O2. Okay, that NADP and ATP will have to be taken to that of stroma. Okay, in that stroma, where that it will have to get carbon dioxide, water is present in that stroma. It is a matrix, so that water is present here. So in this way, CO2 and H2O and NADPH, ATP, these are the assimilatory power. What are they are called? Assimilatory, that is power. Okay, that means NADPH and ATP that are being given to that of stroma, and in that stroma, okay, and in that stroma, that means a dark reaction occur, and this is the part of light reactions, and this is the part of dark reactions. Okay, dark reactions. So dark reactions generally occurs in stroma. Okay, in stroma. That is the first point. What you are going to discuss today, that is about that of dark reaction. Okay, dark reaction. That a dark reaction, you students, one you know that light reaction, another one is a dark reaction. You might have understood that dark reaction is occurring in night, but that is totally wrong. Okay, that is misnomer. That is not true at all. Okay, dark reactions occurs just after that of light reactions, because light reactions provide NADPH and ATP to that of and whenever NADPH and ATP available, whether it is daytime or night time, that dark reaction will start. Okay, dark reaction start just after the light reaction, and it has been proved that suppose there is no sunrise, that is there is no sunshine. Okay, there is no sunshine, so that means NADPH and ATP, that means suppose light reaction is going on, suppose light is not available, that means plant can produce NADPH and ATP, but for sometimes that photosynthesis will continue, and that part of photosynthesis is called that of a dark reaction. After light reaction will provide the byproducts by which dark reaction will continue. And light reactions occur in that of thylakoid membranes and that of stroma lamella, whereas dark reactions occur in that of stroma. Okay, where dark reaction occurs, answer will be stroma. And the dark reaction just occur after that of light reaction, because the light reaction will provide byproducts for continuation of that of a dark reaction. Whether it is a day or night, it is not a matter. But dark reaction has given scientists of that period, and it has been continued. It is being that because you know that is in sunshine on the daytime, that light reactions going on, and that NADP and ATP produce, and so that whatever the NADP and ATP available to the plants, and it will continue that process, and that process may also occur in that of night times. So going to that region, that name has been given the dark reaction, but that name is misnomer. It is not a correct. Okay, it is not a correct of giving that reaction occurring in dark. That reactions also occur in that of light. So it is wrong to call it is a dark reactions. Okay, and it is a misnomer. So now you'll have to know about that. Another thing, you know that dark reaction occurs just after the light reactions, and it uses the byproducts of light reactions, and dark reactions occurs in that of stroma regions. Okay, in that of matrix stroma, that dark reactions occur, because water, it will have to get water, and carbon dioxide it will from the outside, and NADPH, ATPH are called that assimilatory power, which will be helpful in that of production of that of glucose. Okay. Now we'll have to know that, students. We'll have to know that. I'm just showing you that of just showing you that of two equation. Okay. I'm just showing you that of two equation, and we can have to call me which one is the right equation. You see that 6CO2 plus 6H2O. Okay, gives rise to C6H12O6 plus 6O2. Okay, you mark it. Here is that carbon dioxide and water are needed for the synthesis of glucose. Okay, so that this is also a reactions by which carbon dioxide and water will form that of glucose and also that of oxygen. You mark it. And that another equation, students, you mark that 6CO2 plus 12H2O. Okay, gives rise to C6H12O6 plus 6. Okay, 6O2 plus that is also 6H2O. Okay, 6H2O. You mark it that. Which equation is that of correct? Okay, which equation is that correct? This equation is correct. This equation is correct for photosynthesis. First one or second one? Which one?
Sir, second one. Second one.
Second one. Okay, second one. You see that. You see that. Here you see that 6CO2, that is C6, 6. Then you see that here also that of 12 oxygens, here also 6, 18 oxygens. Here you see that. Here also 12 oxygens, also 6, 18 oxygens. You see that. Here also hydrogen 12, here is a hydrogen 12. But this equation is not acceptable. Because you see that from that of 6, that is 6 water, that means what it will be produced, that will be not molecular oxygen. It will be the nascent oxygen. Okay, it will be that of nascent oxygen. So that will be nascent oxygens like this. And it is unstable and it is harmful. Okay, so that it will produce that of nascent oxygen. If it is 12H2O, it will produce that of that is molecular oxygen. Okay, molecular oxygens. It will produce molecular oxygens, which is helpful both for the respiration as well as for the other organisms and for that of existence in atmosphere also. So that molecular oxygen, it will form that of molecular oxygen. But whenever that is 6H2O, it will produce that of that is 6 nascent oxygen, not that molecular oxygen. That is nascent oxygens, but it will produce nascent oxygens. Okay, that is harmful. That is harmful. So owing to that reason, in photosynthesis, the 12 molecules of water is needed. Okay, 12 molecules of water is needed in photosynthesis in order to produce 6 molecules of that of oxygen. That is molecular oxygen. Okay, that is molecular oxygen. In order to produce molecular oxygens, that water molecules should be 12 in numbers. That is a concept in that of photosynthetic reaction that you have to remember. Why there are 12 molecules of oxygen instead of 6? That answer is that to get that of molecular oxygen, to that of 6 molecules of oxygen, that is molecular oxygen. If you have to use that of 6H2O, you will have to get that of 6O2, that is nascent oxygen. That is not a molecular oxygen, that is nascent oxygen. And also water vapor you can't have to get here. Okay, no water vapor you can't have to get here. In the transpiration, that is in transpiration process, that water vapor comes out to the atmosphere, so that will not happen. So that is that authentic. Okay, this is that authentic reactions to that of that is that reaction, that is photosynthesis. So this reaction is available. This reaction is that most important reaction for that of photosynthesis. You see that here that glucose is produced. Okay, here is glucose is produced. Glucose is produced. Okay, that glucose is produced. And that glucose is produced, but glucose is transported, that is transported to other parts of the plant in form of that glucose,
Glycol.
sucrose. Okay, glucose is produced, but is transported in form of sucrose. Why it is? It is stored in form of starch. It is stored in form of starch. It is distributed to other parts through phloem, that in form of sucrose. Why glucose is distributed in form of sucrose, not in form of glucose in plants? The answer is that glucose students, that is what? It is a monosaccharide. It is a monosaccharide and highly reactive. And it can have to make reactions with that of other molecules. But here sucrose is a disaccharide. Sucrose is a disaccharide. So sucrose is a disaccharide. So it is non-reactive. It is non-reducing in nature. I have told you that is disaccharide. In disaccharide, that is sucrose is what? Sucrose is non-reducing. So it can non-reducing. So it can't bind with other molecules, and it cannot be also reactive. It is non-reactive. Non-reducing means non-reactive, and it can also that means it has energy value, high energy value. Okay, its value is higher than that of glucose. Glucose is monosaccharide, but sucrose is that is disaccharide. And disaccharide, you know that it is made up of that of glucose plus that of fructose. So as it is two monosaccharide, so it is highly energy based. And as it is more energy, that means it is the more energy carrier. That means it will have to carry that of it has more energy, and it will have to be distributed throughout the body parts of the plant, as well as non-reducing in nature, non-reactive. So it is so glucose is used in form of sucrose in plants instead of glucose, as glucose is monosaccharide, and that is less energy, and as bearing less energy, whereas sucrose is disaccharide and bearing more energy. And glucose is reactive, whereas sucrose is non-reactive. Glucose is reducing, whereas sucrose is non-reducing. Owing to that reason, sucrose is used. Okay, that is sucrose is used as food materials, and it is being distributed as food materials throughout the body of the plants. Okay, but it is reverse in case of animal. In animal, sucrose is not used. That is, sucrose is not used. Glucose is used because you see that sucrose can be converted into glucose in plants due to the presence of enzyme sucrase. Due to the presence of enzymes, what? Enzyme sucrase. Okay, but that enzyme presence in our body is very less. Okay, so sucrose cannot be converted into glucose. Our body can accept that sucrose, that is sucrose, it can, that means sucrose will be given. That means in plants sucrose is given, but that sucrose is converted to glucose also. That glucose by means of that of sucrase enzyme. So sucrase enzymes are adequately mostly present in that of plant cells, in plant, but it is not available in that of animal. So their availability is less. So going to that reason, you are using glucose, not in form of sucrose. Okay, in animals, that is glucose is used because lack of sucrase enzymes, and even that is sucrase delivery, delivery channel, that is process, is not also found in that of animal body. That means such transport systems for that of sucrose transport system is also not available in that of in that of animals also, where it is adequately available in that of a plant. So sucrose is used in animals, whereas glucose is used. Sugar in form of glucose is used in animals, where sugar in form of sucrose is distributed, is available in that of a plants. Okay, students, that you must have to remember that of why sucrose is used, why glucose is used in form of sucrose. Okay, why sucrose is used in that of plants and then not in is used in plants and not in animals. That concept is very important and we must have to remember that one. Now we come to that of dark reactions. That of dark reactions, you know that is I have already told you that occurs in stroma. Okay, that is in matrix stroma. It occurs in stroma. And the second point is that with the product of that of ATP and NADPH, which are that of assimilatory powers, and also take that of carbon dioxide from outside and water, so that it produce that of glucose. So that glucose, so that sugar is synthesized, carbon, that is carbohydrate is synthesized. So going to that region, it is also called biosynthetic phase. Okay, it is also called biosynthetic phase. Why it is called biosynthetic phase? Because carbohydrate is synthesized in this phase, in the dark phase. Okay, going to that region, it is called biosynthetic phase. That is food is synthesized in this reaction. So that is called biosynthetic phase. And it is also light independent phase. Light independent phase. Okay, that is a light reaction. That is light dependent. It is light independent. Okay. It is a light independent phase. Light independent reactions, okay, or that of phase. That is biosynthetic phase. Dark reaction is known as biosynthetic phase because glucose is synthesized in this phase. Second point, it is light independent phase because because this reaction does not require light, but it depends upon that of products of light reactions. Okay, it does not depend upon light. No light, sun rays is required in such reaction, but the products of light reaction is very much important for running this reaction. So it depends upon the products of depends on light reaction depends. Okay, depends upon depends on the products of light reactions. Okay, and it is start, that means dark reactions continues for sometimes even if light reaction stops. Okay, light reaction stops. Continues. Dark reaction continues for some times, even if light reaction stops. Suppose light reaction stops, that means that dark reactions also not independent, that is immediately stop. It will continue for some times because the byproducts of that of light reactions, that is ATP and that of NADPH, that are also used because that are available. Okay, dark reaction will continue as long as NADPH and ATP are available. So you have to write down another point also. Will continue till NADPH, DPH and ATPs are available. Okay, are available. ATPs are available. So that you have to remember that dark reaction is called biosynthetic phase. It is a light independent phase or reaction. It depends on the products of light reaction. It continues for some time, even if light reaction stops. Continue till NADPH and ATPs are available. Okay, so these reactions also continues. Also goes on in night also. Okay, so there is no question of that. It is a dark reactions and it is held in that it is goes on in night also, and it is also, and that dark reactions also else in daytime. In daytime. Okay, also helps in daytimes. That point you have to remember, that of dark reaction. Now you'll have to know about that of dark reaction consist. This is very important. This is the anyone can have to remember. Okay, so that dark reaction consists of that of three cycles. You'll have to know that these three cycles, friends, these three cycles are very important. Okay, you have to remember, and you have to remember conceptually. That means what are these three cycles? Okay, and you will have to know about that of three cycles. So that first one, and I have told you that that is dark reactions. Okay, you know that that is a dark reaction. In that of dark reactions, that is a oxidation process, and that of a reduction process. In that of okay, that point you have to remember that that a dark reaction is a reduction process. It is a reduction process. Dark reaction is a reduction process. Have you any idea that that is what are that of oxidation process? That is removal of a hydrogen is called oxidation. You know that that is removal of a hydrogen is called oxidation. Addition of a hydrogen is called reduction. So hydrogen ions are added. Okay, hydrogen ions are added in that reactions. So it is called a reduction process. But light reaction is a oxidation process. Okay, light reaction, photooxidation, that is, you know, that photolysis of water, that is photolysis of water, where hydrogen ions come out. That is hydrogen, that removal of hydrogen, that is proton, that is removal of hydrogens. So that is called that what? That is called oxidation process. Whereas here hydrogen ions are added in formation of a glucose, so it is called that of a reduction process. Okay, now we'll have to know about that of a dark reaction, cycles in that of a dark reaction. That dark reaction, students, has a three cycle. One is that of a C3 cycle. Okay, it is otherwise known as Calvin cycle. Calvin cycle. Another one is that of a C4 cycle. This is otherwise known as, students, that is Hatch and Slack cycles. That is Hatch and Slack cycle. Okay, Hatch and Slack cycle. Then we will have to know about that of a CAM cycles. Okay, another one is that of a CAM cycle. CAM, C-A-M. CAM cycle. Okay, that is called Krasnodarb's acid metabolism. That is called Krasnodarb's, that is acid metabolisms. Okay, metabolism. Generally, it is in the glycolysis. It is found that up, and it is also found in that of angiosperms. In angiospermic plants, such process is that is glycolysis, glycolytic path, that is Krasnodarb's acid metabolisms. Okay, that is important that you have to remember. And that is another one, that is C3 cycle. It is a universal one. Okay, that means whatever be the plants, photosynthetic plant, in the all photosynthetic plants, that Calvin cycle or C3 cycle is present in all plants. Okay, in all plants. So this is universal in nature. That is universal in nature. Okay, the formation of glucose is the main objective of that of Calvin cycles. Okay, Calvin cycles. And that C4 cycle and that CAM cycle also, they have also that of similar objective, but they are little bit different from that of Calvin cycle. So Calvin cycle is universal in nature. It is found in all plants, that is C3 cycles. And now we'll have to read about that of a C3 cycle, that is Calvin cycle. Okay, dark reaction means you can have to remember that is very easy. Whatever points I have told you, that using a products, no misnomer, that is calling it is in dark reaction. That is not the important factor. Important factor is that of that means which phases. That is also called free cycle or Calvin cycle or Calvin that is hashtag cycle, or that is hashtag pathway. Also, it is also called of that of that is hashtag pathway. Two scientists, they have found it, that is C4 cycle, and that is CAM, that is Casselson's acid metabolism, that is CAM cycles. Okay, these are the three cycles. One will have to know one after another. Okay, and now we'll have to discuss about that of Calvin cycle or C3 cycles. Okay, that is C3 plants. There are two types of plants also, C3 plants and that of C4 plants. Okay, you'll have to know about that of and first you'll have to know that is what are that of that is what is C3 cycle and what is Calvin cycles. What are the objective of the Calvin cycle, and how what are that of different okay stages of that of Calvin cycle. Okay, students, now we'll have to know about that of Calvin cycles. Okay, Calvin cycles. Before that of study of that of Calvin cycle, students, I'm just telling about that a few concept of organic chemistry. Okay, organic chemistry. That is few concept of organic chemistry. It is very important for you that you see that. That what I am telling you, that is most important. You see that. First one, you will have to come to that of alcohol. Alcohol. That is alcohol after oxidations. Okay, after oxidations, what it is becoming? It is becoming alcohol to that of aldehyde. Okay, that is aldehyde. And aldehyde after oxidations becoming that of acid. Okay, that is oxidations. That is oxidations. Okay, and reverse is that that means when acid comes to that of acids, come to that of reductions, come to that of aldehyde. Okay, aldehyde, that is reductions, and that aldehyde also after reduction becoming to that of alcohol. Okay, that is alcohol, that is reductions, that is reduction. So alcohol after oxidation, that is becoming aldehyde, aldehyde in that oxidation becoming acid, and it is reverse in that of acid becoming aldehyde in reductions, and that of aldehyde also come to that of alcohol. Okay, have you know that of glycerol? Suppose you have to take the example of that is glycerol. Okay, that is glycerol. I have to, if you have to, anyone asks you to draw the glycerol, how can you have to draw? That you see that CH2OH. Okay, that is CHOH and that of CHOH. Okay, you see that you have to draw like this CH2, CH and the CH2, and you have to make that of a bond. Okay, carbon. That is glycerol. And the glycerol after, that is after oxidation. Oxidation, what it will be, students? It will be that of a glycerol aldehyde. Okay, and it will have that of a, that is glycerol aldehyde. Okay, aldehyde. That is glycerol aldehyde. And if you have to ask that of a draw, that means what? How you have to draw? You see that. That is C double. That is C and the C. Okay, that is CH2OH. Okay, CH2OH, and we'll have to draw that of OH also, and that is OH, and that is hydrogen, that is OH. Here also it's okay. That is of the glycerol aldehyde. It is due to that. You see that. That is oxidation O double bond here, so that it is due to that of a glycerol aldehyde. And then glycerol aldehyde, if you have to, then more oxidation, what you will have to, that is glyceric acid. Okay, that what you will have to get, that is glyceric acid. Okay, that is glyceric acid. We will have to get that of glyceric acid. The last one, what you will have to get, that is glyceric acid. You see that it's, you will have to form that of OH, also to that of OH, and that of O. Okay, that is glyceric acid. What you will have to get, that is glyceric acid. Okay, that will be double bond. Here is double bond, so double bond. That is glyceric acid. So glycerol after oxidation becoming, that is glycerol aldehyde. Glycerol aldehyde after oxidation becoming that of glyceric acid. That is glyceric acid, also known as that is also called that of glycerate. Okay, that is glycerate also. That glyceric acid also called that of glycerate. Okay, that you have to remember. That means how oxidation, that alcohol becoming aldehyde, aldehyde to that of acid, and in that of reduction, acid becoming aldehyde, aldehyde to that of alcohol. So if you have to take the example of glycerol, that glycerol after oxidation becoming that of glycerol aldehyde, glycerol aldehyde also after oxidation becoming to that of glyceric acid, and that is called glycerate. Okay, and this is that of that means if you have to on the draw that of on that of basis, you can have to draw that CH2OH, single bond CHOH, CHOH, and that is a glycerol aldehyde. C, that is double bond O, single bond A, C, single bond OH, that is alcohol. You know that is glycerol aldehyde, and that CCH2OH, that is signifying that of aldehyde. And you see that is glyceric acid, OH, 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 and double bond O, that is about that of glyceric acid. That is the common general idea that of organic chemistry. That is a concept of organic chemistry that is some way attached to that of that is Calvin cycle. What you are going to discuss about that of Calvin cycle. Okay, that is Calvin cycle. Calvin cycle. Okay, now I'm just discussing about that of Calvin cycle. Okay, that is Calvin cycle, students. That is Melvin Calvin. That is the name of that of scientist is Melvin Calvin. That is Melvin Calvin, and his colleague, that is colleague name is that of that is Russians, and Melvin Calvin. That is Melvin Calvin. He is the scientist and his colleague. Okay, and his colleague that is Russians. Both in cooperation, okay, discuss about that of cycle in that of dark reactions, how glucose is formed, so that means he conducted an experiment by using that of a green algae. Okay, by using that of a green algae, that is chlorella. Okay, that means he took a green algae, that is green algae means that is chlorella. Okay, that is chlorella. In chlorella, that is a green algae, Okay, that means he took a green algae, that is also photosynthetic plant, and in that green algae, that means he used that of 14C radioactive, that is 14C. Okay, 14C radioactive carbon, that is radioactive carbon isotope. Okay, isotope. He used that of 14C radioactive isotope of carbon. Okay, radioactive isotope of carbon, that is 14C. That means you know that carbon has that is 12C, but instead of that he took that of radioactive carbon isotope, that is 14C. Okay, 14C. In this process, he observed that of that is photosynthesis process going on in that of chlorella. Okay, in that of chlorella, a green algae, and that experiment was conducted in year 1957. In which year that experiment conducted? In 1957, and he observed that means students, you see that I'm just telling you that is CO2 plus dash is equal to 3 molecule of carbon. That means can you feel this graph? That means you see that here is a 1 carbon. Here you can have to add how many carbon, so that you can have to get 3 carbon. Generally 2 carbon. Yes, generally 2 carbon. 2 carbon, generally 2 carbon, because that chemically, that means that is 1 carbon in carbon dioxide. Here is 1 carbon, and if you have to add 2 carbon, you can have to get a product that is called 3 molecule carbon. Because Melvin Calvin found that there is 3 carbons. Okay, there is a stable product. That means he used that of 14C radioactive carbon isotope process. Okay, he used that process in which he observed that photosynthesis in that of chlorella green algae. He found that the first stable product which is coming from that of photosynthesis is a 3 molecule carbon. So owing to that reason, it is called as that of a C3 cycle. Calvin cycle, that is Melvin Calvin and his colleague Parsons. That means they belong to that of California University of that of United States of America. They observed a chlorella, a green algae, photosynthetic process he observed in that of chlorella by using that of 14C radioactive isotopes. And he that experiment offered, that is in 1957, he found that a stable product coming from that of photosynthesis in chlorella is not is a 3-molecule carbon. It is a 3-molecule carbon. Okay, students, that is 3-molecule carbon. So that what is that of substrate? So what should be the substrate by which carbon dioxide will have to make reaction to produce that of a 3-molecule of carbons? That means all the scientists said that that is 2 carbon, but such experiment found that there is no such carbon found in that. But they found that there is a RUBP. Okay, there is a carbons. Okay, that is a 5-carbon molecule. That is a 5-carbon molecule, students. RUBP. That is ribulose 1,5-bisphosphate. That is 5-carbon molecule. That is 5 carbon. Here is 1 carbon. You see that how it is 3 molecule of carbon. That is a controversy. Okay, you must have to be surprised if it is not 2. If it is not 2 and if it is 5, that is 5 carbon molecule, that is ribulose 1,5, that is bisphosphate. I'm just writing that ribulose 1,5. Okay, that is bisphosphate. Okay, that means ribulose 1,5-bisphosphate. Ribulose in the sense that it is related to that of ribose. Ribose and the deoxyribose, they are pentose sugar. Students, I have told you that ribulose, that is some way similar to that of ribose that are used in that of DNA and RNA, that is deoxyribose. So they are pentose. So they are 5 carbons. Okay, how many carbons? 5 carbon. Ribulose is 5 carbon. 1,5-bisphosphate, students. You see that. That means phosphates are on that of 1 carbon and on 5 carbon. Okay, there are 2 phosphate. There are bisphosphate in the sense means 2 phosphate group. These phosphate group are added at 1 and 5. At 1 and 5 carbons. First carbon, then 5th carbon. On that of ribulose, phosphates are attached. Going to that region, they are called RUBP, ribulose 1,5-bisphosphate. That is RUBP, student, stands for
Ribulose 1,5-bisphosphate.
bisphosphate. And how many carbons are in RUBP?
5 carbon.
5 carbons. How many phosphate groups are in this that is molecule? How many phosphate?
2.
2 phosphate. Very good. 2 phosphate. And at which carbon they are attached with that of ribulose?
1 and 5.
Yes, very good. 1 and 5. That means at 1 and 5. Okay. That means you can have to, if you have to see it, you can have to easily tell. Okay. That ribulose is it is a sugar. That is 5 carbon sugar. That is pentose sugar. And 2 phosphate groups are attached. At which carbon they are attached? 1 and 5. Okay, easy. So that RUBP. So RUBP, that is CO2. So carbon, when carbon attached to that of RUBP, that means they will produce 3 molecules of carbons. Okay, they will produce what? 3 molecules of carbon, students. Okay, that is important. Okay, that is important that we will have to know about that. Okay, that is 3 PGA. Okay, that what they will have to produce, that is PGA. 3 PGA you will have to produce. Okay, now I'm just telling about you and showing that of Calvin cycle. Have you remembered that? That is Calvin cycle was first of all who discovered that of Calvin cycle?
Sir, Melvin Calvin. And his
And his
colleague
colleague name is
Basam.
Bassam. Okay. And they belong to which country?
California.
California. Which country?
Sir, America, United
United
States.
States of America. In which year such experiment was conducted?
1957.
1957. And which green algae that means he used for the experiment?
Chlorella.
That is chlorella. Okay, chlorella. Very good. And that means which carbon isotope that was used? That is 14C. Okay. That is 14C carbon. Okay, that is 14C carbon. Now I am drawing that of cycles. You also mark it. Okay, it is very important. I am just drawing and you also draw out simultaneously. Okay, that is, I am just drawing. You see that, students. You see that what I am drawing, and you will have to draw simultaneously with me. Okay, that you see that. Here is that. You see that, students. You see that here is that of carbon dioxide. That is one molecule of carbon. Okay, one molecule of carbon is being attached with, okay, is being attached with RUBP. Okay, that is ribulose 1,5, that is bisphosphate. Okay, that is 5C. That is 5C. That is 1C. Why I will write? And that means when that of carbon dioxide will have to, that means we'll have make reaction with that of substrate RUBP. What it will produce, students? They will produce that of, okay, that is 2 carbon of that of PGA. Okay, that is 2 carbon of, that is 3 carbon PGA. What it will produce? Phosphoglyceric acid, that is 3 PGA. Okay, 3 PGA. And that means which enzymes also, that is Rubisco, enzymes. Okay, that is Rubisco enzymes. We know that which enzymes will have, that is Rubisco. Okay, Rubisco, you know that is ribulose, that is bisphosphate, carboxylase and oxygenase. Okay, that is enzymes. And I have told you that means which is the most abundant protein in this world? That is
Rubisco.
Rubisco.
This is also that most abundant enzyme in the world also. Okay, most abundant enzyme as well as protein in this world, that is Rubisco enzymes. So that Rubisco help in that of transformations of that carbon dioxide and RUBP converted into that of what? That is 3-PGA. You will have to, that is phosphoglyceric acid. Okay, that is phosphoglyceric acid. That is glyceric acid. How many carbon atoms? They have 3 carbon. 3 phosphoglyceric acid. That is 3 carbon. Here is 5 carbon. Here is 1 carbon. So it might be 6 carbon. So how it is 3 carbon? Because you see that means 2 molecules of PGA will be formed. 2 molecules of 3 PGA will be formed. As it is 3 carbons, so 3 PGA, so 2 molecules of, PGA is formed. You see that means 2, 3, 6, that is 6 carbon. You see that 5 carbon, 1 carbon, 6 carbon. Now you see that equation, 6 carbon. You see that means here that is bisphosphate. I have told you that there are 2 phosphate, but you remember that 3 carbon will have 1 phosphate. So here is 3 PGA, that is phosphoglyceric acid, that is 1 phosphate. So where it's gone? Another phosphate. You see that phosphate is also present, because 3 PGA will produce 2 molecules, 1 3 PGA plus another 3 PGA. Okay, so that you see that RUBP has 5 carbon. Carbon dioxide, that is 1 carbon. If they will meet together and will have to produce a product, that first stable product. Okay, that is the first stable products. First stable product of photosynthesis. So what it will have to get? That is 3 PGA. 3 PGA, 3 carbon. Where has gone another 3 carbon and another phosphate? So you see that they will produce 2 molecules of 3 PGA. 2 molecules of 3 PGA. So 2 molecules I have given food bracket. Then on the outer part I have written 2. That is 2 molecules of 3 PGA. In this 2, 3, 6, 6 carbons and 2 phosphate. That is 3 carbon, 1 phosphate, 2 molecule, 1 molecule carbon. That is 3 carbon has 1 phosphate. That is 2 molecule of carbons. That is 6 carbons. How many phosphate? That will be 2 phosphate. Now equation ready. Okay. And these students, this process is called carboxylation. And this process, this is the first process of the Calvin cycle is carboxylation. That is called carboxylation. Okay, carboxylation, in which carbon dioxide, you know that carbon dioxide is added in which process? That process is called carboxylation. Okay, carboxylation. So after that, this is the carboxylation process in which carbon dioxide meets with that of REPP, that forms that of 2 molecules of 3 PGA. 2 molecules of 3 PGA. 3 PGA is 3 carbons, so 2 PGA, that is 6 carbons. Okay, and it is the first stable product of photosynthesis. But it is not always the first stable product. 3 PGA is not first stable product for all plants. That means for which plant 2 PGA is the first stable product, that plants are called, that is 3C plants. Okay, and those are that of that is oxalic acid, oxaloacetic acid, OAA, that is 4 carbons, they are called, that is 4 carbon plants. I will tell you later on, you will have to learn it in gradual process. You know that is first stable product, students. Then after the PGA, you see that, then what will happen? The second reaction is, students, you see that 3 PGA will be that of 3 will be, okay, but that will be diphosphoglyceric acid. Diphosphoglyceric acid. That will be told as, students, what? That is 1,3-diphosphoglyceric acid. That is, it will be 1,3-diphosphoglyceric acid. Okay, you see that. That is 1,3-diphosphoglyceric acid. You can have to write down in a small letter also. Okay, that is 1,3. So it will have to break into that of 1,3-diphosphoglyceric acid. That means this one becomes that of di-2. Okay, phosphoglyceric acid. You mark it that. That means you see that here are that is phosphor. That is, you see that here is 3 phosphoglyceric acid, but here 1,3-diphosphoglyceric acid. That means how one phosphate group added with it. In that case, that is ATP. That is ATP will be broken, and it will be ADP plus PI. That PI is inorganic phosphate. That inorganic phosphate will add with that of 3 PGA. So it will become 1,3-diphosphate, that is phosphoglyceric acid. Okay, it will be 1,3-diphosphoglyceric acid. That is the second step. Okay, second, and then 1,3-diphosphoglyceric acid, students, will be, that means that will be in a reduced forms, and that will be phosphoglyceric aldehyde. Okay, that will be that you see that process, the second process, this is carbogylation. This process is called, the second process is called that of reduction. In that of reduction, 1,3-D-PGA will become that of PGAL. Okay, that is PGAL. You see that how from acid becoming that of aldehyde. Okay, phosphoglyceric aldehyde. That is aldehyde. That is PGAL. That is also that of triose. Okay, that is triose phosphate. Okay, this one is also called that of triose phosphate. This one is called that of triose phosphate. Okay, that is called also triose phosphate. And that is phosphoglyceric aldehyde. Phosphoglyceric aldehyde. That means on reduce. That means how it is reduced. That means when hydrogen will be added with that of 1,3-DPGA, so that there is also breaking of that of NADPH. Okay, that means also broken into that of NADP plus H plus. Okay, that H plus go to that of added with it, so it becomes that of when that is reduction. When hydrogen will be added, that will be reduction. So that is a reduction. The second process, first process is that is carbon dioxide addition, that is carbogylation. Second process is reduction. Reduction means hydrogen ions added with that of DPGA. Okay, diphosphoglyceric acid. When phosphoglyceric acid is added with that of hydrogen ions, that become what? Aldehyde. Okay, that become aldehyde. So that is PGAL, and that PGL also, students, is that of 3 carbon. Okay, that is triose phosphate. This is also 3 carbon. Okay, triose phosphate. This is also 3 carbon. 3 carbon, and that triose phosphate will have to what? That is produced. The next step is that of a glucose. So here glucose is synthesized. That in reduction. In reduction, that NADP is converted into that of NADP plus H plus, and that hydrogen ions added with 1,3-DPGA and becomes that of PGAL, and the reduced form of that of reduced form of acid becoming that of aldehyde. I have told you that of simple concept of organic chemistry, how acid becomes that of aldehyde. Okay, and you see that if there will be no regeneration, then how it will be a cycle? So there will be a regeneration. Then next process is, student, is that of regeneration. Okay, third process is that of regeneration. How it will be regenerated? You see that here is RUBP. RUBP has 2 phosphate, that is 5 carbon, 2 phosphate. Here you see that. Here you see that is PGAL, that is 3 carbon. So in this case, that is triose phosphate, and a glucose is formed. That glucose is formed. It is pentose sugar. So it is pentose sugar. You know that glucose is that of that means you see that what it will be. Glucose has also, because here is RUBP, that is pentose one, that is 5 carbon. So it will have to take that of ATP. What it will have to, if it will have to break ATP here, you see that here also ATP breaks into that of what? That ATP, and just here ATP that breaks into that of what? That is ADP plus PI. Okay, and that PI mixed with that of PGL. Okay, PGL. So that here is that you see that. Here you see that. Here are only that of what? Here is RUBP. So that means phosphate. Here is 3 carbon means 1 phosphate here. 1 phosphate. So 2 phosphate are required for RUBP. So if ATP will have to be converted into that of ADP, and then that is phosphate, inorganic, that is phosphate. You see that is ATP. So that phosphate will be added with PGL. So there is a 3 carbon. If phosphate group will be added, that will be how many? That you see that will be, that is phosphate group added. So that will be 2 phosphate group. So that RUBP. So that PGL, after becoming adding with that of phosphate, 1 phosphate group, that is becoming that of RUBP. Okay, RUBP. So this is that of cycle, Calvin cycle. In Calvin cycle, what it is telling that first process is that of carbogylation. Okay, first process is the carbogylation, in which RUBP is being addition with carbon dioxide comes into that of 2 molecule of 3 PGA. That 2 molecule of 3 PGA after addition of phosphate group becoming to that of 1,3-D PGA, and that 1,3-D PGA, that is diphosphoric, that is phosphoglyceric acid, after addition of hydrogen becoming reduced, and that reduced form of acid is that of aldehyde, and the aldehyde will become the RUBP, because there is a 1 phosphate group is here, So that ATP will be broken into that of ADP. That is ATP will be what? That is ADP and that of PI, and ADP and ADP and that of PI. So that phosphate will have to meet with that of PGL, and it will have two phosphate groups. So that will be RUBP. Okay, RUBP. So this is that of a Calvin cycle. In that of Calvin cycle, you see that. That means you see that here are two molecules of PGA, so two ATP. So two ATP molecule will be broken. Here is, you see that NADPH. Here also that of what? That is a diphosphoglyceric acid 2. So 2 NADPH. So 2 NADPH. So 2 NADPH. So that is another ATP. That is 1 ATP. So you see that for one carbon, so for one cycle, for one Calvin cycle, how many ATPs are required? You see that. That is 2 ATP, 3 ATP are required, 3 ATPs. How many NADPH required? 2 NADPH are required. Okay, 2 NADPH are required. You see that. That is carbon dioxide. Carbon dioxide, that is 1 carbon. And for glucose, that is 6 carbon or hexose. So 6 carbon are required. If you have to synthesize glucose, you have to, that means you have to make how many Calvin cycle? How many tons of Calvin cycle? For one carbon molecule, there is a 1 ton of a Calvin cycle. For 6 carbon molecule, how many tons of Calvin cycle will be, students?
For
6
6.
ton.
How many?
6 ton of carbon cycle.
Okay, 6. So 1 Calvin cycle is, that is 1 carbon molecule has 1 Calvin cycle. 1 carbon molecule, for 1 carbon molecule, 1 Calvin cycle. Okay, that is 1 Calvin cycle. If you have to produce glucose, how many Calvin cycles are needed? 6 Calvin cycles, 6. So 6 Calvin for glucose, for production of 1 molecule of glucose. How many ATPs will be needed? That is 3 into 6, 18 ATPs. How many NADPH will be required? Into 6, that is 12 NADPH. Okay. So that for 1 carbon molecule, that is 1 Calvin cycle is needed. For 1 Calvin molecule, that is for 1 carbon, that is 1 Calvin cycle. For synthesis of glucose, 6 carbon is needed. So there will be 6 Calvin cycle. For the 6 Calvin cycle, how many ATPs will be required? That is 3 ATPs for 1 Calvin cycle. For 6 Calvin cycle, it will be 18 ATPs. For that of 2 NADPH required. For that of 6 carbon cycle, that is 12 NADPH. So for synthesis of glucose, how many ATPs are required? 18 ATPs are required. For synthesis of glucose, how many NADPH is required? 12 NADPH is required. Okay, students. So that is about Calvin cycle. Whatever I have left, I will have to tell in that of evening part. Okay, in that of evening session, evening class, that I will have to tell you about that of Calvin cycle, and another one is that of sheep-4 pathway. That is also that has like pathway. Okay, and then I will have to make tests. I have already told you that that is 5 pages. You must have to read, and I will have to test that per class bio test that will be conducted in the evening sessions. Okay, students, that is another class is there. So thank you very much for joining. Thank you, students.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.